To complete our discussion of the carbon cycle, let's take a closer look at some of the things that can happen to organic carbon once it has accumulated as plant biomass or net primary production. The proportion of net primary production that is eaten by herbivores differs dramatically among open water, submerged, and terrestrial ecosystems. In open water environments, the majority of algal biomass is consumed by herbivores, typically about 70 to 80%. Algal cells are small, bite-sized, and they can reproduce fairly rapidly, so herbivores can mow down their food source and still have a steady source of NPP. This also means that algal biomass does not tend to accumulate in open water ecosystems over time. Most of the carbon ends up as consumer biomass. In upland terrestrial ecosystems or in emergent marshes, typically very little of the vascular plants gets eaten. These plants are typically larger than the things eating them, structurally complex with large inedible parts, and often have anti-herbivore defenses. This also means the plant biomass does tend to accumulate in these ecosystems. Submerged macrophyte communities are often intermediate, with about 30% of NPP getting eaten by herbivores. There are some exceptions. Some terrestrial ecosystems, especially grasslands, can support high rates of herbivory, and plants can continue to resprout from their root system. It also helps if the herbivores migrate through the ecosystem over time, giving patches of plants time to recover. For example, some crabs have been found to devegetate large areas within salt marshes, but when allowed to move freely, these patches will typically regrow, and the crabs will move on to other grass patches over time. Soil carbon is a reservoir we haven't really discussed yet, but soils are an important area of carbon storage. Inputs to soil include senescence of plant biomass, including litter fall or accumulation of dead root mass below ground, and cellular exudates that plants release from their roots. These include organic acids that they release in order to access soil nutrients more easily. Processes that remove carbon from soils include leaching to groundwater, or fragmentation and decomposition, or respiration of soil organic matter by soil microbial communities. Let's take a closer look at some of the components of the soil community. Bacteria are a major component. They're extremely diverse communities. For example, this community on the left side of the slide shows the diversity of bacteria contained in a 5 milliliter or 1 teaspoon sample of soil. They also perform most of the chemical transformations in soil ecosystems. They're responsible for turning particulate organic matter into dissolved organic matter, which makes nutrients more available and also increase rates of leaching from soils into groundwater. We will take a closer look at some of the bacterial reactions and some of the chemical transformations later in this lecture. Fungi are important parts of the soil community as well, particularly in stressful environments with dry, nutrient-poor, or acidic soils. They can tolerate acid conditions. They also aid in weathering and decomposition of soils. And for that reason, they're often found in symbiosis with plants because they can help plants to get access to soil nutrients that would otherwise be unavailable. Fungi within soils are very extensive and they're often very abundant, forming very large networks below ground. Animals in the soil community aid in the physical breakdown of organic matter, from large pieces to particulate or dissolved organic matter. They often free nutrients and dissolved materials for further use by bacteria or by fungi. The main factors affecting rates of decomposition of organic matter in soils include litter quality, or the quality of the material that's being broken down, temperature conditions, water availability, and oxygen conditions. Different chemical components of organic matter decompose at different rates. Labile substances like cell solubles and cellulose decompose relatively quickly. These are relatively small molecules with regular structures, so many organisms have the enzymes to break them down, just not most animals. Cell solubles are what you use to make tea. They dissolve easily in water, and they are among my favorite forms of dissolved organic carbon. Larger, more complex molecules, which we collectively refer to as lignins, decompose very slowly. Most organisms do not have the enzymes capable of dealing with these molecular structures, so they tend to accumulate in the soil. Also, one more thing, notice the differences in time scale on the x-axis between tropic and Arctic ecosystems. We'll come back to environmental conditions in just a bit, but the rates at which material break down are related to quality, but also to local environmental conditions. Organic matter tends to decompose in an exponential fashion. Check out the equation in the upper right of this slide. You may notice that it looks similar to the exponential growth equation that we would use to estimate population growth. Only this is a decay function, so we're measuring a decrease in abundance rather than an increase in abundance. We use the constant K to express the intrinsic rate of decay of the material. A high K will mean a faster decay rate, and a low K will mean a slower decay rate. We can quantify decay using leaf bags. We would put a known mass of material in a bag, leave it out in the soil or bury it in the environment of interest, and collect subsequent measurements of mass to see how quickly the material is lost from the bag due to decomposition. The quality of the litter is related to the value of K. So this graph shows the decomposition constant K on the y-axis and the ratio of lignin to nitrogen in the leaf on the x-axis. Notice that leaves with low, excuse me, need some more dissolved organic carbon. Notice that leaves with low lignin and high nitrogen decompose quickly, while leaves with high lignin and low nitrogen decompose more slowly. It's worth noting that litter quality is in part determined by the amount of nutrient available in the environment. Plants growing in high nitrogen soil will tend to have more nitrogen in their leaves. When they shed those leaves, they will tend to decompose more quickly. So adding nutrients to an environment will influence how quickly organic matter breaks down. At a single location, the rate of soil respiration has a monotonic relationship with temperature. The rate of decomposition will increase with temperature, just like any chemical reaction. Beyond that optimum, the organism's proteins may denature or cease functioning properly. Across many locations around the world, the relationship between decomposition and temperature is more linear or increasing. The warmer the location, the faster organic matter will decompose. This relationship is a consequence of local adaptation of the soil community to local conditions, such that they are optimized to function at the temperature where they live. Oxygen is a key determinant of decomposition rates. Aerobic respiration is the form of respiration that yields the most energy. So organisms that use oxygen to decompose organic matter can break down organic matter more efficiently. Oxygen can become low in wetland soils. Water limits diffusion of oxygen into soils, And the rate of use of oxygen by respiration can outstrip the influx of new oxygen. When oxygen is used up, organisms must use anaerobic forms of respiration, which yield less energy and are therefore less efficient. Acidity from organic matter can also limit the growth of bacteria, especially in wetland soils. Wetlands are among the most productive ecosystems per area in the world. The net primary productivity in swamps and marshes rivals that of tropical rainforests. Some of this organic matter accumulates as peat. When it becomes dissolved, carbon is also exported to downstream ecosystems. So wetlands can be an important source of carbon. Organic matter tends to accumulate in soils wherever rates of production are greater than rates of decomposition. If decomposition is high, then soil organic carbon does not accumulate, even in really productive ecosystems, for example, tropical rainforests. If decomposition is low, soil carbon can accumulate, even in relatively low productivity ecosystems like the tundra or in bogs. Grasslands represent one area where organic matter tends to accumulate. Dry conditions and seasonal cold inhibit decomposition. In addition, grasses produce a lot of biomass below ground in the form of roots and rhizomes. So a lot of organic matter starts out below ground in the soils. But by far, the sites of the greatest soil carbon accumulation are wetlands. And among wetlands, bogs accumulate the most. Bogs have all of the qualities that inhibit soil respiration, including cold, anoxic conditions or low oxygen, acidic soils, and more features, which we will return to in the next video. This photo shows a bog in Europe that is being harvested for fuel. You can see that the peat accumulation can extend meters below ground. A core that we extracted from a fen in the Altona Flat Rock contained more than 4.5 meters of peat and included organic matter that was more than 6,500 years old. Take a look at this heat map of soil carbon content. Areas that appear in green and yellow represent areas of highest carbon accumulation, and red areas represent areas of lowest carbon accumulation. You will notice hot spots in carbon accumulation in cold regions, particularly in the northern hemisphere and in alpine or mountainous regions. And you will also notice hotspots in wetlands, including northern peatlands, but also riverine wetlands, even in tropical regions. Terrestrial ecosystems tend to get the most attention with respect to carbon sequestration. Blue carbon is a movement to acknowledge and measure the importance of coastal wetland ecosystems in storing carbon. I've included a video on the course page below. Take a look and think about the questions I've posted on the course page. We will return to this topic later in the course when we discuss the role of wetlands in global climate change.